Hi, my name is Hana Warmflash and I'm from Portland, Oregon. Today I'm going to explain the process known as polymerase chain reaction, or PCR for short. PCR was basically created to mimic cell or DNA replication in cells by isolating a specific region of the DNA and amplifying it by making lots and lots of copies of it. Now, there are three main stages in PCR that rely on the usage of different temperatures. But before we get into these stages, we need to know what the components of our PCR master mix are. That will be our DNA template, our forward and our reverse primers, our nucleotides, our DNA polymerase enzyme, a buffer that goes along with it, and water. Once these are all mixed together, the test tube that contains it can be put into our thermal cycler, which will raise and lower the temperatures for us as needed. So our first stage is the denaturation stage, which is at the highest temperature, up at 94 degrees Celsius. At this stage, it essentially melts the hydrogen bonds between complementary strands of DNA, giving us two single strands of complementary DNA. Then we can lower the temperature way down for our next stage, which is our annealing stage. And this is where the engineered primers come into play. They're attached to their complementary strands of DNA that directly precede the region of interest that we want to amplify. These are really important because free nucleotides can only attach to pre-existing strands of DNA, so they need their primers. And then the temperature is brought back up sort of towards the middle for our final stage, which is our extension, which is where the enzyme, the polymerase, attaches onto the area of the DNA that has the primer, and it pulls the free nucleotides and extends the DNA sequence all the way down until it reaches the end. It follows our direction of synthesis from our 5' to our 3' end, depending on our phosphate and our OH molecules, which always run in the same way. This enzyme is super special in a PCR reaction because it needs to be able to withstand extreme temperature fluctuations. So we get ours from the hot springs in Yellowstone National Park. We get it from the bacteria that lives in those, known as TAC, so we call it our TAC polymerase. After each cycle of the PCR, we have twice as many strands of this DNA, meaning that after each cycle, and we have 35 total, we double the number every single time. So in the end, we have billions of copies of DNA, which can be used for further analysis or further sequencing and reveal a lot of information.